Heavy metal contamination is a common source of soil pollution caused primarily by mines and smelters. The main metals involved in this contamination are mercury, lead, and arsenic. One promising method for remediating high concentrations of these metals in soils is mycoremediation. Mycoremediation is a form of bioremediation where mushroom mycelium, the main body of a fungal colony, concentrate and pull contaminants such as metals out of impacted soils. It is a very promising technique because it is potentially cheap, effective, and environmentally friendly. Many studies have investigated the effectiveness of using mushroom mycelium to remediate heavy metal contamination. For example, Singh and Srivastava found that a particular strain of fungus was able to reduce arsenic levels in a soil sample by almost 82%. Another study found that mushroom mycelium was able to remove 85% of lead from a sample in highly acidic conditions with a pH of 4. In practice, mycoremediation has been used in a water contamination project in Newtown Creek, New York, in which rings of fungal material were sent floating around, absorbing heavy metals from the polluted waterway. For our experiment, we used oyster mushroom mycelium to try to reduce levels of heavy metal contamination in soil samples. To assess this effect, we sprouted lettuce seeds in containers with one of two treatments. In the first group, they were sprouted in plain contaminated soil from the Opportunity Pond Superfund site, and in the second group, contaminated soil was mixed with 1/3 mushroom mycelium. These containers were watered daily and left under 24-hour light for 3 weeks. After this time, the seedlings were counted, removed, and the height of each seedling was measured along with the total weight of the seedlings from each group. From this, we found that many more seedlings sprouted in the mycelium-treated soil. However, the contaminated soil sprouted taller plants on average, which is not what we expected. This suggests that the mycelium treatment may increase the number of seeds that successfully sprout, but may have a negative effect on seedling height. We suspect that if the seedlings were allowed to grow for a longer period of time, they would outgrow the contaminated soil seedlings. If we had more time and resources to repeat this experiment, we would grow the seedlings for a longer period and attempt to precisely measure the levels of contamination in each treatment. Our soil remediation group would like to thank Tony, Michelle, and the helpful staff at Planet Natural for making this project happen. Thanks for watching.